Going. I've got the quizzes to give back at the end. You've got homework due, you've got a quiz due, and then recitation is pretty much tied up. Next week in recitation, bring questions. I'm going to try and do some sort of review over the whole semester, which is going to take a while, but we have the full 50 minutes. We have an exam coming up week on Monday. I believe it starts at 10:30 and it goes through till 12:30. Good chance to catch up, good chance to do well. And let's see how we do. I'm talking about peptides, and I'm talking about amino acids and things like that. Things that some of you, well, most of you will have to deal with in the near future. And the bond is basically an amide. You're looking at making amides here. So you can think about how we would do this in the lab. We might take an acid chloride, we might treat it with an amine, maybe with a base or something, and then you get an amide bond. And those things are quite strong. We've said that they are typically the sort of goal in terms of a synthesis is to get there, and they're not typically used for other things. They're not typically used as start materials for other materials. But we can see now the sort of choice here. You can't use a carboxylic acid to make a biomolecule or a conjugate because it doesn't have another R group at the end. You can use an ester. When we get into polymers next week, we'll see that polyesters are very important, and also polyamides. So to finish this off in terms of the peptides, I just want to go back a second and talk about the problems with these things. The idea that organic chemists try to mimic nature, we try to make naturally occurring materials and then modify them to see if they have different properties. And the problem with this type of thing is it's complicated by the fact that you have multiple functional groups and you have to protect. So in this case, we've taken a very simple system where you just have two different R groups, A and B, if you like, and you think about how you can put those two things together. They both have amino groups, they both have carboxylic acid groups. So they could, like a cross-aldol or a cross-Claisen reaction, react with each other or react with themselves. And in that case, you're going to get four different products, and that's a complicated mixture, and that's not very useful synthetically. So what organic chemists have to do is block and protect. And this costs money, it takes time, and it's laborious, but it has to be done. We can see now if you took two amino acids with two different R groups, and don't forget the R groups might have something attached that needs to be blocked, that complicates it even further. You can mask one end and mask the other end over here so that you only leave one carboxylic acid open and you only leave one amine open. Then you can come up with some sort of system to dehydrate. That's the condensation idea, where you take a molecule of water out of here. DCC doesn't mean anything to you. If you take 5821 in the fall, it will, dicyclohexyl carbonate imide. And you'll find that that's a very useful coupling reagent to be able to dehydrate systems and give an amide, right? In this case, a dipeptide. And you keep doing this. You take off the blocking groups and you go again. This molecule now is a dipeptide, and you'd have to maybe put together another blocking group to stop it reacting with itself, and then you can add a third material and so on. So the types of groups that we use are fairly straightforward. If you want to come to listen to some of the talks this afternoon, anybody who's interested in the master's program, our students are giving their first year talks at 3 o'clock in Ward Beach from 6030, and everybody's welcome to come watch these things. You'll see that some of the chemistry that my student Chris is talking about is carbamate chemistry. You can see here a different functional group that you really haven't seen yet, which is the carbamate, nitrogen, carbonyl, oxygen. And that's the basis of his thesis work, so you might be interested in that. But these types of compounds are good temporary blocking groups for amines. And it turns out amine chemistry and amine functionalization or protection is actually a big deal. It's actually quite tricky to do. But these compounds have been made useful. You can see here a big UV active group. You can see this on a TLC plate very easily as you're using it. And we have this temporary blocking group that we can then get rid of. So in terms of doing this, we can think about how to block the amide or the amine end, and you can think now of using something like this, a simple acid chloride that produces the carbamate, and that's robust enough to not react under the conditions that we're going to use for synthesis. How do you block a carboxylic acid? Well, I think most people will be happy with the idea that a very simple way of doing it is to make an ester. Yeah, the carboxylic acid proton is acidic. If you want to get rid of that temporarily, turn it into an ester, because you can turn it back into the carboxylic acid either by saponification or by acid catalyzed hydrolysis. So we can set up now a complicated system in which we're trying to bring several different amino acids together, and this is done on large scale in the medicinal chemistry industry, pharmaceutical industry. And you can see the peptide at the bottom, the polypeptide here is insulin, and if you're trying to make this or trying to make analogs of it, this gets to be quite laborious. Again, starting out with two amino acids, we have to block both of them, we then have to do a couple, but that puts two of them together. I can see about 35 in that molecule, so that takes a long time. But one of the big things that's happened in the past 30 years or so is the automation of these types of ideas. You can now do these with machines, you can do these with robots, you can program the synthesis, and we have synthesizers that you can actually program to add, like PCR. Many of you have done PCR probably, or heard of PCR, polymerase chain reaction, where you put your amino acid mixture in, your nucleic acid mixture in, you set it to a certain temperature and you let it go. That's similar to what we do now with amino acids. So you can actually program these things, and it's much easier than it used to be. But it's still laborious. Now, I'm not going to talk about this. This is heading into biology, but that's basically the relationship there. You've got to have nucleic acids to code your proteins, and you've got to have proteins to code your nucleic acids. It's the old chicken and the egg argument. How does this happen? Well, in terms of forming a peptide in nature, you conjugate one of the amino acids to, in this case, a hydroxyl group on a ribosugar, and then you do this. And what mechanism does this remind you of? Again, this is cheating. This is kind of lazy, but that's the way they've done it. What mechanism is that? Nucleophilic acyl substitution. And when you do biochemistry, and you do maybe my class next spring, you'll find that nucleophilic acyl substitution is probably the biggest mechanism that you see in biological processes. It's very, very, very prevalent. So we can see now this is doesn't require blocking groups because everything's set up in a much more orderly manner, and we only get one outcome. So that's how we make peptides in nature. Now, in terms again of where we're at, I'm here, I'm happy here. You will have to be happy there, and many of you will have to be happy there. You think about the bigger subjects like pharmacy and medicine and what have you, you have to be comfortable and conversant in all those different languages. Now, somebody asked me this the other day, Kimball, and the question was in recitation, I think. It was something about grapefruit. I don't like grapefruit. Nothing personal, but I just don't like them. Apparently, you can't take grapefruit with certain medications, or you can't take certain medications with grapefruit. This was the question. And, you know, I went home and thought about this and asked my wife what the problem was here. And we find out that actually they contain certain organic chemicals that screw up some of the enzymes that are required to break down the pharmaceuticals. And a lot of the medicines that we deal with are non-natural compounds. They've been made in the lab synthetically, and the body is doing its best to destroy these things, to break them down. Especially after it's done its job and it's found its way to the liver, and the liver's whole purpose is to detoxify stuff for the most part. You have to try and break these molecules down into small components that can then be excreted. Well, the problem with grapefruit here is that it contains these compounds, and you're all familiar with these compounds. There's a fairly simple natural product that comes from grapefruit. So you can see an aromatic cycle in the middle. You can see a lactone, an ester at the far right. You can see an alpha-beta unsaturated system. You can see an aromatic furan ring at the left. You know a lot about those types of compounds. What shape do you think they are? Approximately. Flat. Yeah, they're flat. And for some reason, they inhibit the activity of something called cytochrome P450. We've mentioned that a little bit in the past. These types of enzymes in the liver that are used to oxidize organic material. So let's say you had some toluene. Remember what toluene was? Methylbenzene. It's used as paint thinner. You can imagine if somebody ingested this stuff, it could be toxic. But it makes its way to the liver, and the liver has to try and get rid of this stuff. So what does it do? It oxidizes the methyl group to a carboxylic acid. Now, we've done that in here, right? We've done that with sodium dichromate. It's a similar type of idea where you have a metal type system that wants to be reduced, and the carbon gets oxidized, and the whole point of making that carboxylic acid is to make it polar. And once you make it polar, you can get rid of it in the bloodstream. So you can imagine now that this is a problem because the grapefruit contains this type of compound, and if you're trying to get rid of a poisonous metabolite of a medicine, or you don't want this to build up and become toxic, cytochrome 450 has to work. So now we can see this is the structure of that particular enzyme. That obviously is a huge, great superstructure. And in here we have a substrate. You'll see a lot of these things in the near future. But the problem is that grapefruit contains compounds that block the action of this enzyme. The compounds find their way into the active site and block the action. So they can't detoxify materials. So that's just an example of a polypeptide. That is a huge polypeptide that happens to be something that you'll see in the future, and it answers Kimball's question. Is that okay? There you are. Now, I just brought some pretty pictures. This means nothing to me. This is a whole bunch of nice ribbons and strings and things, and it's pretty. But it's HIV protease. And again, the translation from my class were detail, 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 every atom counted. All of a sudden now you just don't have time to do that. So you'll have to get used to this idea of using these ribbons and these types of sheets and twists and things to be able to show the activity of proteins. You'll see that you do have to see the ligand, or the piece that is actually operated upon. Often that will be detailed because you need to see that. But again, this is where you have to go, many of you. Some other examples, alpha-glucosidase. What's the simplest example of a glucosidase? Anybody know at this point in your lives? Anybody chewing gum? Anybody planning on eating today? Salivary amylase. Okay, salivary amylase is a protein that takes apart carbohydrates. And if you go back to the carbohydrate material, you recognize that they're all held together by acetals. And acetals are stable to base, but what do they do in the presence of acid? They hydrolyze, right? They hydrolyze back to ketones or hemiacetals or whatever you want. So this is essentially, again, the equivalent of H+. I'm happy with my H+, there's your biological H+, good luck. Okay, that's the difference. Other examples of this, methyltransferase, you will see that You have to methylate various things. If you look at the nucleic acid material we skimmed over, taking uridine without the methyl group at 5 to thymidine, which has the methyl group, the big difference between RNA and DNA as far as the bases are concerned, it requires a transferase. Guess what transferases do? They transfer things. Now that's the easy part of biochem. They transfer things. And this is going to be used to do essentially the same thing as methyl iodide. You'll find there's a big cofactor involved called S-adenosylmethionine, which has a really fancy leaving group attached, which is the equivalent of methyl iodide. So you'll be able to see that in the future. And this is the protein that modulates that type of process. Other examples, C-lectin here is just a protein that binds carbohydrates. That's going to be a big deal in the future if anybody's interested in medicine or pharmacy. People are starting to understand the interactions between carbohydrates and their receptors. And if you can block those types of processes, they could have inferences in anti-cancer compounds, stuff like that, in terms of cell recognition. So keep an eye on these molecules in the future. They are now very, very useful targets for medicine. Vitamin D binding protein looks fancy. Again, I don't see any chemistry in there, I don't see any atoms, but you'll have to get used to what all these beta-pleated sheets and these barrels mean in the near future, and you have to really be on top of stuff because that gets quite overwhelming. So that's about it for amino acids. I just skimmed over it. To me, as an organic chemist, an amino acid is an amide, or it contains an amide bond. The peptides contain amide bonds. Amino acid contains a carboxylic acid and an amine. We've done both of those types of chemistry in the near past. We should be okay with that from now on, I think. So the one I want to finish off with in this particular section, which is a summary of 24 through 26, is the third type of molecule. And if you look at the title at the top, the way I organized this when I put it together was chemistry and water, which is biology basically. And you can see the amino acids at the top right are very polar. They do contain polar material, so they should, for the most part, dissolve up in water. Nucleic acids, likewise, they should be comfortable in water because they have lots of hydrogen bonding possibilities on the different functional groups. Over the left, we certainly will expect carbohydrates to dissolve in water because they're very, very polar with all these hydroxyl groups. Now you move to lipids. Give me another word for lipid. Anybody have any idea what lipids, what are they? Fats, right? So typically, you think about the historical use of animals as food stock materials. The fat is non-soluble, and the way you isolate it is to let it rise to the top of boiling water, and you'll skim this off, and then you have fat. So it's non-polar. We can see here that lipids are typically, they don't contain many functional groups. That should be interesting. As you head into biochem, many of you, you've got to worry about how these things are made, the biosynthesis. And the problem is you can't do biosynthesis without functional groups. So I'm going to lead you through this towards cholesterol. We're going to make cholesterol between now and Monday, biosynthetically. And you'll find that, just like organic chemistry, biology needs functional groups. But at the end of the day, those functional groups get lost. They're used to build a system, and then they're taken out. An awful lot like what we do in the synthetic lab. So in terms of the types of system you've probably heard of to get this started, at the top you have a triglyceride. Okay, so a triglyceride, obviously there are 3 of them, so tri. It's unsaturated because it contains a double bond. And in that case, what is the stereochemistry of that pi bond? Z, isn't it, or cis. And if you take that and you hydrogenate it, and you then end up with a saturated fat, you completely change the structure of that molecule. This molecule has a flat region, and it also has a stereochemically defined region around that double bond where the 2 groups are pointing in the same direction. If you head to the saturated fat, you lose that, and you end up with a system that can rotate freely, so the molecule changes its shape dramatically. And there's all sorts of background to this in terms of the health consequences of unsaturated fats versus saturated fats. A lot of it's down to molecular shape. So to get this started, I'm going to talk about the synthesis or the biosynthesis of cholesterol, which is a lipid-type compound. And we're going to find that it summarizes a lot of chemistry that we've done this term. So when you get into biochem or further classes like this, you'll recognize that organic chemistry is really the foundation of that. Go back and read the syllabus from the first semester. This material sets up that type of subject. Organic chemistry is central to a lot of different things. So what we start off looking at is something called acetyl coenzyme A. Okay? And if you think about this, it's got this very, very complex looking nucleic acid component. It's held together through a phosphate linkage, and it has this material pantothiene, which you'll see later, as a linker. And at the end of it, it has this, and there's the acetyl component. So it's a thioester. It's not a regular ester like we would see, but it's a thioester in which the O has been replaced by S, but it's the same family. And what you'll find is that all this is just a fancy leaving group. There's a lot more to it than that. The shape helps it bind to a protein, so the protein can fish it out of a solution and stick to it to enable to do its job. But this is just a fancy leaving group. And if you want to think about an equivalent in the chemistry that we've dealt with, that whole molecule up there is the equivalent of that, acetyl chloride. Obviously, you can't use that in nature, right? You wouldn't recognize it. There's not enough material there to stick to. And also, if you produce HCl, that's probably not a good idea. So nature uses this type of thing, where the sulfur is the leaving group. Now, why should sulfur be a better leaving group than oxygen? It's large, right? It's bigger. All the way back to the first couple of days of class, the first couple of weeks of class. Now we're heading into a bigger subject, but it's the same principle. So this type of functionality now is going to be used as a 2-carbon piece to be able to knit molecules together to go from 2 to 4 to 6 to 8 to whatever's needed for the biological system to form its lipids. At the bottom, I have a cartoon or a sort of picture that shows the utility and the versatility of this type of system. And it's all about alpha carbon chemistry. You should be spending time in 22 now, and of course 23, getting yourself happy with that stuff for the final, because there is a significant amount of it on there. But again, this is central to biochemical construction. We can see here that if we stick with this structure, we can use the carbonyl carbon as an electrophile, and nucleophiles can go after it. If that nucleophile is another carbon piece, you can make carbon-carbon bonds, which we will do in a minute. And then you could imagine this. Now, what is this process called? Tautomerism. That is exactly right, tautomerism. Tautomerism is everywhere in biochemistry. Got to be aware of it. And you can see now that if you form the enol, you can switch the roles of the material. On the left, it's an electrophile. On the right, it's a nucleophile. And this comes back to things that we did with Br2, the Hell-Volhard-Zielinski reaction, the whole point of the aldol reaction. You can make both the nucleophile and the electrophile from the same material. There's an example from biology. So you see this a lot. Nucleophiles come in, you get nucleophilic acyl substitution. In this reaction, you get electrophilic addition, both of which we've studied in great depth. So I'm going to start off and work our way through this. And the first thing I'll show you is that each of the reactions in this biosynthetic process, which is quite complicated and you will see again in the future, is a simple organic reaction that you have seen individually. The problem is you now have to put it all together, as we kind of have been doing for a couple of weeks now. So in the first reaction, we have this material. And that should look familiar. We have 2 carbonyls, 1 and 3 atoms away from each other. And in this case, it's a little bit modified to what you've seen. This is a biological system that we don't use in the lab. Instead of making an enolate, because really what this is serving as is this. What we need for this Claisen condensation, if you remember, is some leaving group and a negative carbon. That's the nucleophile on a Claisen reaction. But what's the problem? What's the pKa of a typical ester? What's the alpha carbon proton in a typical ester? You will need to know these things. Not just now, but later. What is it? 25, right? So it's not very acidic. So what do you need to take it off? You need a strong base. Well, obviously, if you're in aqueous medium, you don't have hydroxide. You don't have super strong bases, and you usually don't have super strong acids. So nature has developed a way of getting around that. Instead of taking a proton off and dumping a negative charge at that alpha position, it uses this type of system instead. Now, what you'll find is kind of fun about biochem is that this is all occurring inside a big protein, or at the surface of a protein. These molecules have been brought together, they've been sequestered, they've been organized. In organic chemistry, it's very difficult to get 3 molecules in a row. It's very difficult to get molecule A attack molecule B at the same time it's attacking molecule C. That's very difficult. There's nothing there organizing the system. But in biology, you have proteins. The proteins bring the pieces together. They hydrogen bond to them, or they have electrostatic interactions holding them in place, and all of a sudden you have a template. And that template now is used to do these types of reactions that we can't do in the lab. So imagine again, this is inside some sort of cave, right? It's all been brought together. It's all pre-organized. And what's happening here is a decarboxylation. So the byproduct is carbon dioxide. And we'll follow these arrows. This forms carbon dioxide when this bond breaks. Now, typically, we've pushed the electron density into the carbonyl when we did the decarboxylation reaction, which you are okay with now. But what if you have a carbonyl close by? That's already pre-organized right next to the material that you're using as the nucleophile, and we have a nucleophile able to attack an electrophile. Now, I haven't finished this. What's the next arrow I should put on this system? 2 electrons up onto the carbonyl, right? And there will be something close by that stabilizes that. maybe a proton, maybe a carboxylic acid that will then stabilize that negative charge as it starts to increase. But then we've got a leaving group attached. So what mechanism will happen now? What mechanism will ensue? You have a carbonyl with a leaving group. What's that? There are no tricks here. Right, nucleophilic acyl substitution. And you'll find in biochemistry that often it's the simplest answer. There's nothing tricky here. There's nothing funny here. So we'll think that this electron pair can come back in, this can break off, and then what we have is this, and that looks an awful lot like things we have made. That is a beta-keto ester, very much like what we made from a Claisen reaction. In fact, that is a Claisen condensation with a modification because you can't make a negative enolate because it would be way too basic. Well, that's almost, there's a useful structure. You have knitted 2 carbons together to 2 and made 4, and you now have to go further and you have to start thinking about modifying the backbone. We don't need that hydroxyl. That hydroxyl has to be lost. We don't need that ketone. That ketone has to be lost. And now it comes down to very simple carbonyl chemistry where we have to modify this using ideas that you're familiar with. In the laboratory, what reagent would you use for that? Look at the starting material, look at the product in this next step. What would you use? You have an ester and you have a ketone, and one of the pieces is becoming an alcohol. What would you use? A selective reducing agent. Do we have one in our toolkit? NABH4, right? So again, a lot of you are thinking of pre-pharmacy, a lot of you are thinking of medicine, a lot of you are thinking of things in the health industry. This biochemistry is medicine, basically. So be careful with this stuff. This will, if organic chemistry didn't get you, biochem might. So be on top of stuff and be seeing the patterns, seeing the different ideas. NADPH is a biological equivalent of borohydride. Does exactly the same thing. It introduces H-. You'll see that. So what we did there was reduce the ketone. Why reduce the ketone before the ester? Why does that make sense? Esters are stabilized by resonance where ketones are not. So the ester is a little bit less reactive, and even in the lab you would expect the ketone to go first. That's fairly straightforward. Now, we don't want that hydroxyl. That hydroxyl does not show up in the backbone of these lipids, right? They're trying to make one of these chains by adding these pieces together again and again and again. But that hydroxyl needs to go. So what should we do? What should we make it behave as? A leaving group. Make it leave. In the laboratory, what would you do? You could add some acid, and expect it to do elimination. Or you might add a tosyl group and then add a base, and maybe it'll do something like that, eliminate. And that will give you a pi bond. So we'll see as we work our way through here. A very similar type of process happens, and then we end up with this. And this is an alpha-beta unsaturated ester, very much like the stuff we've been talking about recently. So I now have a bifunctional molecule. I have a system which has a delta plus carbon here, and I have a delta plus carbon here. So in the last step of this, to get to a alkyl chain, you need to reduce and get rid of that pi bond. In the laboratory, what would you use? Think of the final here. Did we learn anything? H2 and platinum, absolutely. H2 and platinum, H2 and palladium. Doesn't work in living systems, obviously. But again, we have this type of system. We have a reducing agent. And chemically, this is a little bit beyond my class, but it's a measure of where you're at. If you don't get this stuff or this stuff turns you off, don't do biochem. Right? Go do something else. But think about this chemically. This NADPH is the equivalent of H-. So where do you think it attacks in this molecule? Look at the product. We brought 2 hydrogens in. One of them's here, and one of them's here. Where do you think the H- attacked in our alpha-beta unsaturated system? The 4 carbon. Yes, the beta carbon. Just like it would if it was a cuprate. Just like it would if it was a malonate type system in the Michael addition. That's what's happening. You bring in H-, then you develop a negative charge, and it gets protonated with H+, and you have a reduction. So that's kind of clever. So what we're going to do here is try and use this stuff to go up to bigger compounds and ultimately cyclize some of these things and end up with cholesterol. And this is fun. Again, I would, in all seriousness here, if this is boring, don't do biochem. Don't go into pharmacy, don't go into medicine, because that's all it is. If you enjoy this stuff, great, because that's what you're going to get more of it. Thinking about the uses of some of these lipids, you have these types of molecules where the N here is quite long, and it's usually units of 2 for the same reason we just looked at. You're bringing in 2 plus 2 plus 2 plus 2, you get these multiples of 2 in these units. So looking at these membrane anchors, you might find that these things are very nonpolar here and very polar out here. So you can imagine that these chains are going to find their way into nonpolar environments, such as a cell membrane. so they can anchor themselves into a cell membrane. Then you have functionality sticking out of that cell membrane that could be used for further conjugation. Now I'm going to get rid of that noise and ask you what this is. What type of structure is that? It's a quaternary ammonium salt in a biological system. We've already seen a little bit of phosphates. We've obviously seen a lot of esters, typical simple functional groups that are now building up into very complex molecules that help us live. I've shown you this before. We can go back at this now and look at the whole aggregation idea. This is a whole bunch of carbohydrate. So this is the red material right there. The blue stuff is the water. Should make sense now. Big molecule, polyhydroxylated, wants to find the aqueous environment because that's where it's comfortable, that's where it can dissolve if you want. Down at the bottom, all the gray material, this stuff here, long fatty acid chains. They're going to find themselves away from the water and aggregate with each other. So they organize themselves in these lipid bilayers and these cell membranes, some of which you've probably seen in the past. You will see a lot of that in the future. So I want to get into some biosynthesis. This is the type of stuff we're doing in my other class. If you like this stuff, take the class I teach next spring, 2016, 2 semester hours, 2 hours a week, a lot of fun. Examples of lipids that are very important in biochemistry and just basically keeping you alive. Prostaglandins. You may or may not have heard of these things. They are organic chemicals that have 5-membered rings with long fatty acid chains attached. And they serve all types of purposes. Some of the most obvious ones or most important ones are they are actually ligands. They bind to proteins called GPCRs, G-protein coupled receptors, which send signals in and out of cells. So these ligands initiate processes in a cell. And you can see here some fairly straightforward organic structures. I can see a carbonyl. I can see here, what type of alcohol is that? Secondary. What's the pK of this thing approximately? 16. If I was to bring in a phenyl group, a magnesium type reagent, some sort of grignard, it would go for the ketone. What's the pK over here? About 5, right? And if you were to bring in some acid and some alcohol, you might make an ester over here, you might make an acetal over there. Simple organic materials. Well, nature makes them from this. And what's the problem down here? We don't have any functional groups. So nature has to install them. You get oxidations, you get reductions, you get hydrations by adding water to things. Very simple things that we have seen. Well, to get started on making these things, let's go back all the way to the beginning. Let's go back to the basic building blocks, which you will learn in great detail in biochem, to be able to put together bigger pieces. And I'll tie this into next week, which is about polymers, right? All the idea of polymers and putting groups together so that we can make bigger systems very easily, very nicely to make plastics and things like that. These types of reactions, are you listening up there, engineers? You're just all bored today. You might as well go home or play with your phones and take the class again. It's entirely up to you. Children. Okay, so we're going to make these basic building blocks. And we're going to start thinking about how this works, the whole idea of leaving groups, the whole idea of nucleophiles, and the whole idea of coupling these things together in a very simple, very logical way in a biological system. Other examples that these things go into to build things like vitamin A, things like beta-carotene. They all contain these 2-carbon building blocks which you add up again and again and again to make these much bigger systems. So this is where you need to be. And biochemistry is an interesting mix of detail, where you see bonds being formed and broken, and just big pictures and big schemes, where you don't see a chemical. And this is the first example of that. This is the biosynthesis of cholesterol. And we start out with acetyl-CoA, which is the 2-carbon piece we dealt with a little while ago. And you start building these things up. And you start building bigger pieces, and bigger pieces again, and bigger pieces again. And you end up with much bigger pieces, which then get to the central building block, farnesyl pyrophosphate. And that thing can go in all sorts of different directions to make all sorts of different products. And we can see now that we'll deal with something called squalene, something called guanyl pyrophosphate, and ultimately you'll end up with cholesterol. It's a massive, massive machine type of process to build something that's very important, such as cholesterol. So anybody who's interested in pharmacy probably has heard of these things or has dealt with these things in the past. Statins. Any biosynthetic process that has been nailed down is then a target for medicine. It's a target for biological intervention by pharmaceutical chemists, not pharmacists. They don't do the making of the things, they sell the things. The organic chemists build the medicines. So then you've got some targets. If you know what the proteins are that do the different steps, you've got the possibility of creating something new that could get involved there and interfere, and that could become a medicine. So statins are a very obvious one here that lower cholesterol. Why do they lower cholesterol? They interfere with the process of this. If you can't make that, you can't make the rest of it. So it lower the levels of cholesterol by stopping the biosynthesis at a certain place. That's how it works. In terms of the chemistry here, we're going to take it in chunks. I plan on spending the rest of today and then Monday on this. It takes a while. I've got 15, 20 slides left. So we need to start thinking about how this happens. We need to start building the smaller pieces to build the bigger pieces, and then we'll build cholesterol next week. First reaction. Think about your final. What have I said about the final? I want to know if you learned anything, not just how to play with your phone. Can you put these things together? If you can, good luck in the future. You should do well. If you can't, it's not going to be no fun. Acetyl-CoA. Building a bigger piece here. We already built that, acetoacetyl-CoA. That was the product from the first Plasma reaction from the first earlier slide. And now we're going to couple these two pieces together. And we can see here that we make a carbon-carbon bond, and we end up with a bigger molecule. So in terms of what's happened here, any ideas? It looks like an aldol reaction. It certainly does. It looks like some nucleophile has gone after this carbon, and this carbon seems to be this carbon. So you need to make some sort of nucleophilic component here, and then go after that. Where do you think this carbon dioxide came from? Water kicking out the sulfur. Yeah, nucleophilic acyl substitution. So in terms of how this works, we need acetyl coenzyme A. Again, organic chemists will abbreviate it like this because we don't care about all this stuff. The biochemists do, because that's what makes the molecule stick to the protein to make it work. But that's just a fancy leaving group. So you can do a Claisen reaction. You see something like this 10 days from now in an exam, don't freak out. It's just a Claisen reaction. Just do it. Or an aldol addition. And we end up with this, which is a much bigger molecule. Now we have some functionality. Now we have a hydroxyl group. Now we have a leaving group here for the ester. And we have a carboxylate group, which potentially could come off as carbon dioxide. We've got some pretty intricate sort of materials in there. In terms of what's needed for this, that first step requires this enzyme. All right, a synthase. Nothing fancy here. Synthase are used to synthesize things. Simple as that. Make carbon-carbon bonds. And we can see here that the enzyme is known, the protein is known, its crystal structure is known, and then you can use it as a target. And that's where the idea of the statins came in. The statins are billion-dollar drugs. So these are really useful targets to be able to develop medicines. Now, in terms of the next step, we have to turn HMG-CoA, welcome to acronyms, that's what biochem is all about, to mevalonate. Any ideas what's happening here? Chemically, what's happening in this last step, going from here to here? It's a reduction. If you're in the lab, what would you use? You're reducing an ester to a primary alcohol. What do we use for that? Anybody? LAH, lithium aluminium hydride. You can imagine a question on a final, a 200-point comprehensive final, which is very important to your grade, in which I give you a biological system and say, how do you do it in the lab? Straightforward, just same steps. In that case, what would lithium aluminium hydride do at this end of the molecule? It might also get rid of that carboxylate. So there's where the selectivity comes in. We might not have reagents that are good enough to do this selectively. Biology does. So we can imagine now preserving this because we need it in the next step. And that's the beauty of these systems, is that they're so selective, you can do chemistry at one end and not at the other end and not worry about stuff. So we now have different functionality. We have a primary alcohol, a tertiary alcohol, and we have a carboxylate group. And we go further. Don't forget, the whole point of this is to knit these longer chains together. Mevalonate then gets phosphorylated. Kinases are simply enzymes that phosphorylate molecules. They bring phosphate groups in. And any idea what this will become? What do you think that's useful for? It's a leaving group. That is a really good leaving group. Resonance stabilized leaving group. So phosphates are often used as that. Well, we're almost there. We need to make this into a diphosphate. You'll find that these types of processes enable selectivity. So the system can differentiate between a monophosphate and a diphosphate. So you have some level of control over what's going to happen next. But that phosphorylation is nothing more complicated than the reaction that you did in lab. You did a glucose molecule, you reacted it with acetic anhydride, you made an acetate. The anhydride component, this is now an anhydride, right? And this molecule here is an anhydride. So it's very similar chemistry in which you're transferring one piece to the next. Well, what comes next? Any ideas? What's happening in that top step? An additional phosphorylation. We have added the phosphate group now to the tertiary alcohol. And go back a step. If you did this in the laboratory and you wanted to selectively put the phosphate on the primary hydroxyl and not on the tertiary hydroxyl, how would you run it? It's an addition reaction. You'd use an addition type funnel. You'd probably do put it in a reaction flask and mix it with some solvent. But in terms of temperature, you're trying to maximize the selectivity of reacting at the primary alcohol versus reacting at the tertiary alcohol. What type of temperature do you run it at? Low temperature, right? And what type of idea is that? You're asking the molecule to react at the less crowded place. Is that a thermodynamic idea or a kinetic idea? Kinetic, right? Again, biochem is full of that stuff. So you could do this in the lab. This is done perfectly in nature. So we're going to end up now with a diphosphate, and then we're going to phosphorylate again. And the whole point of this, if you think about the materials we're trying to make here, they don't contain functional groups. We need the functional groups to knit the molecules together, but then we want to get rid of the functional groups. And a good way of getting rid of a functional group is to make it leave as a leaving group. That's what we'll do here. So this carboxylate has been hanging out, waiting to do something. And what it's going to allow to happen is this type of elimination process. Now we've dealt with E1, carbocations. We've dealt with E2, with bases. We've dealt with E1CB, which is also basic. You can't do some of those reactions in nature. The pH doesn't allow it. The pH is fairly close to 7. So you don't have strong bases like hydroxide. You don't have T-butoxide, stuff like that. So methods have been developed here to be able to avoid that. And again, the carboxylate right there is a leaving group, which is allowing the buildup of negative charge. And in this case, what's it behaving as? Give me an idea from an E2 reaction what this looks like it's behaving as. It's something that's breaking off and giving away a pair of electrons. What's that in an E2 reaction, typically? Something small that is grabbed, comes off, and its pair of electrons that it was sharing with the carbon go into the pi bond and become the double bond. Proton. Yeah? That carboxylate group right there is serving like a proton and the base at the same time. So you don't need to worry about using a strong base because this can collapse on its own, and the electron pair has somewhere to go. We talked about the beta-ketoacid parts and the beta-ketoesters. That beta-carbonyl, that 3-keto part, is essential for that decarboxylation to happen. In this case, you can dump the electron density onto the phosphate, which is a really good leaving group because it's resonance stabilized, and away we go. And we have the equivalent of an E2. Gives you a pi bond quite nicely. And so far, we're at this point. Isopentenyl pyrophosphate, IPP. We have now a 1, 2, 3, 4, 5 carbon piece. So we can start to build these things into bigger pieces. 5 carbons, 10 carbons, 15 carbons, 20 carbons, into the bigger terpene systems. So again, nature's quite economical. Why build 2 separate pieces if one will serve both purposes? What does that mean? Well, the aldol reaction. The same starter material serves both purposes. The Claisen reaction, the same starter material can be the nucleophile and the electrophile. And this is similar here. So we have the material we just built. We put a pi bond right there. And we can now understand that the other component that we might need is this. And this is a question about thermodynamics, about equilibrium. Which of these 2 double bonds is more stable? The one on the right, why? More highly substituted, absolutely. But you can get both in an equilibrium. And now you're going to be able to have both to be able to build other pieces. You can see now an abbreviation. This material here has been abbreviated to that. It just has to happen. Biochemistry, it has to happen or else you just don't have time to draw everything. And we have something called IPP down here and DMPP down there. Those are the 2 pieces now, one of which will be a nucleophile, one of which will be an electrophile, with the phosphates behaving as leaving groups, to be able to knit things together. So that was the first step, or the first sequence. We'll start this now and then we'll finish it on Monday, but this is going to go further. And at the end of the day, we'll end up with huge great molecules and this type of deal in the biosynthesis of cholesterol. So we're now going to go from isopentenyl, and don't forget, pent means something, right? What does pent mean? 5, there's a clue. Guaranyl means nothing, but it's 10. And then farnesyl is 15, you keep going from there by adding these pieces together. So here we go. We have a system now in which this 5-carbon piece is coupling with this 5-carbon piece to give us a 10-carbon piece in what looks like a fairly simple process. This piece over here looks like maybe it came from this piece, and then this piece on the left looks like it came from that piece. Any suggestions for a mechanism? It looks to me as if you're coupling this carbon with that carbon to make that bond. Any ideas? We need a nucleophile, you need an electrophile, you need a leaving group. What's the nucleophile? The alkene, good. The alkene is the nucleophile. So you can imagine this thing coming and attacking from there up to there and booting this out. What mechanism does that remind you of? That's an SN2. But this seems to happen so that you put a positive charge here. What rule is that following? You're putting the positive charge on the tertiary carbon, not the primary carbon. What's that? Markovnikov. Now, Markovnikov had no idea about this stuff. He was just interested in simple organic alkenes, but Markovnikov rule still applies here in biology. So you can see how important that stuff has been. So you can imagine now we need an enzyme to couple these two things together. They don't just find each other and do this. You have to have the enzyme close by. You have to have things that will help stabilize the leaving group breaking off, and that then will allow for the coupling to happen. And you end up with a 10-carbon piece. You added 5 and 5, you got 10. So now we have geranyl pyrophosphate, which is bigger, through fairly simple chemistry. Well, then we go further again, and we have to add another piece. Any ideas about a mechanism? Same. This goes after that, boots this out, form a carbocation, lose a proton, and you end up with 15. This is the beginning of polymer chemistry. When we come back on Monday and Wednesday, and I wrap things up with the polymer chapter, that will be the same reaction again and again and again and again and again, up to 1 million times. And then you get long materials that are polymers. So with that in mind, we are pretty close to finishing this molecule off. And we need to take this 15-carbon piece, expand it again, and then cyclize it to cholesterol. And that cyclization process is fascinating. So this part is different. This is beyond the scope of this class. This is why you take biochem or you take my class next spring. This type of system is quite different. The idea now is that 2 of these starter materials come together to produce a dimer. And I'll use that language a lot next week. Monomer is 1, dimer is 2, trimer is 3. This is dimerized. The same starter material, 2 of them have come together to give you a symmetrical molecule in which both of them are represented. And that new bond is right there. The problem is the chemistry here is quite tricky. It isn't simply nucleophilic attack. What has to happen to both of these carbons if we lose oxygen and they end up being bonded to other carbons? If you lose oxygen, what's that? A simple chemical process from high school. It's reduced. Yeah? This is a reduction. And both of those carbons are getting reduced. So there's something much more complicated happening here than we have seen. But again, what's happening is 2 of them are coming together to give this dimeric species. And then squalene. And squalene is a polyene. It has double bonds all over the place. And if you were to add MCPBA or something like that in the lab, you'd epoxidize everywhere. You'd get all sorts of mixtures. So in this case, to get selectivity, it only goes there. How? Magic. No, the enzyme. The protein that's holding it only has that alkene to attack. It's set up properly for that. What do you know about epoxides? Strained, what do you want to do? Open up. So you make squalene oxide, and then you get this. And this is an absolutely incredible process, again mediated by an enzyme, you just don't do this by magic, in which squalene oxide cyclizes. Now I'm going to leave it there so I can give the quizzes back, but that and that is nothing more complicated than acid catalyzed epoxidation opening. So we'll leave it there. Read that stuff, be comfortable with it, ask questions.